AstraZeneca, Bangalore, where he led the unit to deliver AstraZeneca's first anti-tuberculosis molecule for clinical development. And before taking his current appointment at Gangajan, he was mentoring the drug discovery program as a distinguished scientist in various Indian national laboratories under the government of India's Council of Scientific and Industrial Research. And that's a very, very short introduction to VG. He's far more bigger than you can sum up in a few words. His colleague, Mr. Sukumar Hariharan, is also here with us. Thank you for joining. And he's the head of development in GBPL. He has about 18 years of experience in the biotech research. And it is him and his team who have developed the manufacturing process for Actolysin P128, which they shall be talking about. This is their company's lead therapeutic candidate. Mr. Sukumar has extensive experience in the process development of therapeutic bacteriophages, phage-derived antibacterial agents in bacterial expression system, and in their manufacturing for preclinical and early clinical phases of drug development. So thank you both so much for joining and accepting our invitation. We are truly happy to have you on board. And without much ado, may I please request Dr. BG to start.
Thanks, Urmi. Am I audible? You can raise your hand.
Yes, you're audible.
Okay, thank you. Thank you also to the consortium for this opportunity. Like Urmi said, two of us are going to share the talk today. I am more going to talk about how we also started with phages. The question is, how did we evolve from phages to lysins? And this is not just how the company involved, this is more like what science drove, what regulatory aspects drove this thinking from going from phages to lysins. And I'm talking about the path or a voyage which started way back in about 2002 or 2003. So I will talk more on the overview of these aspects, and Sukumar, who has been the project manager, project leader for this project for many years, will be talking about the more nitty-gritty details of the lead compound. Now, before I go into my slides, there are two things I wanted to point out. First is that I'm not going to talk about something which Urmi already alluded to, is the problem of antimicrobial resistance. We all know this. We all know how grave it is. We all know the terrific or the real unmet medical need. We know the current situation where it is rather alarming. So that's one aspect I'm now going to talk about. So we are all taking for granted that we are in dire need of antibiotics, and we've been mostly working with small molecules. Small molecules are around below 1,000 Daltons as the main molecules, which we call as antimicrobials. And today we're going to talk about alternatives. The second point which I wanted to make was that me and Sukumar and me are mostly going to talk about the lead lysin that we have been involved in, which is P128. There are other couple of lysins which are in different stages of clinical development. One which was actually discovered by Professor Vince Fischetti, called X-Becase today. He may touch upon it. The other one is Sal 200, both of which we're not going to talk about. But in general, most of the properties or the way we looked at how these things can be progressed is similar or nearly identical. So if you go to the next slide, so my talk is going to be on the evolution of our thinking. I want to spend a slide on what did we see as our USP, in the sense that what is the technology or platform that we have built, which we believe is what's sustaining us in this area. I'll talk a little bit of P128 as an anti-staph lysin from a perspective of differentiating it as an antibacterial. Lots of antibacterials around. There are new antibacterials for many gram positives. and there are newer ones coming. So how do we look at P128 as a unique molecule to come in? P128, by the way, is about 27 kilodaltons protein. And then a bit on the P128's more challenges as we approach clinics and go through the clinics. Sukumar will take over on P128 details as well as talk about something about other license and our approach. Next slide, please, Sukumar. So if I would look at this in two halves, on the right side is the phage. When we started on phages around 2002, the therapeutic challenge that we found was that everybody said phages carry DNA or multimeric proteins, and you're bringing foreign DNA into the body, so there is a regulatory issue. The other thing was that nobody understood anything about phage pharmacokinetics. Remember, this was also an era where monoclonals had still not become a major therapeutic product. So it's a multimeric protein, it's a protein. How is it going to behave pharmacologically in the human? It's a black box. So that's a regulatory hurdle. Third point was IP for natural phages is difficult. And lastly, as we all know, you developed resistance against phages at a pretty high frequency, 10 to the 4, -4 or -5. So you always need a cocktail. So these are sort of the 4 or 5 points which seem to be hurdles which we couldn't scientifically cross. So we said, okay, what's our alternatives? Let's look at license. Now, remember, we started looking license only around 2007, 2006, 2007. Monoclonals are already, people, it was getting quite accepted as a therapeutic molecule. So license fell into the same category as protein molecules. There were several human proteins in the market around that time. Big help is license do not carry DNA. And of course, generating and protecting IP was a straightforward path. May not be straightforward, but at least we knew how to do it. So this was the contrast that we had to make a decision going from phages to license. Now, if you then reverse the situation and say, today, can I start with phages? We probably look at a more advanced thinking. But anyway, that's what forced us into license. I don't regret that. I'm not trying to say this as saying that, sorry about it. It's just that what drove our thinking from phages to license. Can I have the next slide, please? So license come under the category of non-traditional antibiotics. This is a classification that John Rex has used, and this is more or less being accepted everywhere. He has classified non-traditional antibiotics into 4 beautiful sectors, calls it the STAR, it's called the standalone, transform, augment, and restore, and you have examples for each. I probably will not find to go through all of them, but one of the characteristics of the standalone was that these are molecules which are direct acting. So you have phages coming under that, you have lysins coming under that, you have antisense coming in that. Then you have, if you look on the right side, the transform includes molecules like colistin, which help permeation of other antibiotics and so on and so forth. Now, why am I pointing this out? If you just look at this, this is not differentiating small molecules and large molecules. It's actually differentiating the way they act. So for example, if you look on the right lower quadrant, you have the famous beta-beta-lactamase inhibitor, which is different from the traditional antibiotic, but it's still small molecules. So what I'm trying to say is non-traditional antibiotics includes large molecules and small molecules, but are more classified by their mechanism of action. Can I go to the next slide, please? Sukumar, can I go to the next slide? Okay. I said I will say a few words on our USP. And what we have done over the years is that we either isolate specific phages from the environment, sequence them, or access public DNA sequence databases. And today you have new sequences coming in every few minutes. And one of the things that we have, clearly the lysine molecules that we are looking at is made up of domains. And these domains can be classified into what we call as binding domains and catalytic domains. Now you have a repertoire of binding domains and catalytic domains. You have a library of this. And these binding domains actually let you bind to specific sequences on the peptidoglycan cell wall. And the catalytic domains are enzymatic activities which recognize the sequence. So you can do a lot of permutation and combination, allowing you to combine and optimize activity. And this is how we have built our ectolysin molecule, which we also molecule library. And this is something which we call as our own USP. If I can go to the next slide, please. So some of the unique properties of P128, which Sukumar will talk to you in more detail. And the reason I'm bringing this in is because we're trying to differentiate it as a new molecule. It is a narrow spectrum. So while people look at narrow spectrum as may not be something that the GP will use, but a narrow spectrum has huge advantages in the way we look at the microbiome today. Other striking property of the lysins has been that it synergizes with all the antibiotics that we have tested, irrespective of the chemical class of the antibiotic or its mechanism of action. So clearly, when you see the significant synergy both in vitro and in vivo, what it translates is that you can use very small molecules of P128 so that your doses come down dramatically. Another thing that is important is in many peptides, the biggest problem is when you add it to serum, you lose the activity. This is the right contrast that when you in the presence of serum, especially human serum, there is clearly about 10 to more than 10-fold potentiation of P128. And there are different hypotheses why this is happening. We can talk about it if necessary in the discussion. And lastly, which is I think most important, is that we have seen that if you treat resistant strains, let's say methicillin resistant strains with P128, at a low concentration, P128 will convert that resistance to sensitivity. And this is probably because of its ability to loosen up the cell wall. If I go to the next slide, because I need to hand over to Sukumar right now. 4 points which I wanted to make, which Sukumar will not deal with, but more from a clinical perspective. The challenge has been, how do you position such a molecule? 5-6 years ago, most of the clinical trials that FDA would allow us would then generally be define the infection, right? So we were looking at staph bacteremia with, of course, infective endocarditis. Today, there are more and more trials where you can actually do a pathogen-specific trial. So hopefully that will help us. Then the question is, what's the comparator that we're going to use? And clearly the comparator today is, daptomycin, which is the standard of care. The second point, which has always been an issue to deal with, is are you going to use the lysins as monotherapy, or are you going to use it in combination? Is it important? Extremely important. Because if you're going to use it as a monotherapy, then you're going to show non-inferiority to the available antibacterials, gram-positive antibacterials. While if you're going to use it in combination, the standard of care, then you actually have to show superiority. Superiority obviously is what you would like to show, but superiority brings its own challenges. So this is a question which has always been at the back of the mind when you try and develop molecules like the lysins. The third and fourth points, or the second and third points, deal with P128 being a protein. Are you planning a single dose treatment, or is a multiple dose treatment? In other words, once daily or twice daily for 5-6 days? What happens if a patient who has been treated with P128 comes back 6 months later? Can you actually repeat dose? So, of course, both of them the first one deals with more immediate hypersensitivity, and the second one deals with memory of antibodies which can affect your efficacy. So both these points are clearly something which have to be addressed, and obviously will be addressed during the toxic GLP tox stage. So this is the sort of overview that I wanted to give. I think I've taken 8 or 9 minutes. I will hand it over to Sukumar, who will talk in detail of P128, and then we'll take questions. Over to you, Sukumar.
Thanks, Vijay. Am I audible?
Yes, you are.
Yeah. Okay. Thank you. Thanks, Vijay. So, good evening, all. Glad to be part of this forum where people are looking at Phage and Phage Lysins as a therapeutic. So, common area of interest. As a continuation of BG's presentation, so what I would focus on is Gangajan's journey going from Phage to Lysins, and I would focus mainly on P128, the anti-staph lysine. This topic will mainly cover how did we discover P128 and what are its novel properties, highlighting that, and to summarize where we are right now. So I will not be going into the clinical development part. I'll be more describing on the discovery and the qualities of P128. So all of you are aware of this. So I need to touch upon this because P128 is an ectolysin. So to bring that in perspective, so this is the phage, lytic phage life cycle. So it starts with that adsorption where the phage attaches to the receptors, and then the DNA injection where the phages injects the DNA, and then the third is it's taking over the bacterial machinery and make its own components of protein synthesis, where it's going to make its individual components, and then the phage assembly. So once the assembly is done and the timing is right, then the host cell lysis and the phage release. So this is the typical life cycle. So there are two places where here in this life cycle where the enzymes come into play, that is the bactericidal enzymes come into play. So one is at the entry, that is when the phages are trying to adsorb and try to inject the DNA. There is a role of an enzyme which is involved in bacterial cell wall degradation. So these enzymes are actually tail associated, or the structurally associated proteins, which we call the TAME. And because these are acting from outside, we call them as ectolysin. So the other one is the endolysins, which are phage encoded proteins, which comes at a later stage once the phage assembly is done and the phage is ready to burst, come out of the cell, and there it employs these lysins, which are the endolysins. So the main differentiating factor is phage endolysins are not structural components, whereas ectolysins are structurally are part of the phage structure. So P128 is also an ectolysin. So today's talk will focus mainly on how we discovered P128 and what are the properties with experimentation to support that. So we actually discovered P128 from Phage K. That's basically one of the domain was discovered from Phage K. So Phage K, as all of you are aware, is a polyvalent staphylococcal phage belonging to the Myoviridae family, and it has a genome size of 150 kb. And here is actually the small segment of the genome showing the ORFs from 29 to 67. So as you can see, like for any other phage, the genome of Phage K is also follows a particular pattern. Like all the genes involved in the cell lysis are all bonded together, and the ones which are involved in the structural organization of the phage, like the portal protein, the caps, the tail sheath, they are all clustered together. And we were able to actually find we discovered ORF 56 actually. This ORF 56 turned out to be it had peptidoglycan degrading property. So actually it did not actually fall into the cell, get clustered in the cell lysis among the cell lysis genes. So this ORF 56 is a 90 kDa protein. So when we were trying to understand its property, we were able to go to truncated forms of ORF 56. So we were able to go down from 90 kDa protein to about 16 kDa protein. So basically, these are all N-terminal truncations. So we were able to localize, still have the bactericidal activity to localize to a 16 kDa peptide, which belongs to the C-terminal of ORF 56. And with that, we went ahead. So we also did an experiment to make sure that see that these are structurally associated. So what we did is we had taken the 16 kDa protein, we had antibodies against it. So we had labeled with gold nanoparticles and then incubated with the Phage K, and we were able to clearly see that they were localizing to the tail structure. So indicating that yes, this is indeed a structural protein. And with that, we went to the next step. So basically, we had the ORF 56 from Phage K, and we narrowed down to the CHAP domain, which is actually the 16 kDa domain. And along with that, we had the lysostaphin binding domain, that is a SH3B domain, which is from lysostaphin. So we fused both these together to actually make the protein P128. So it has basically two domains, the catalytic domain from phage K and the binding domain from lysostaphin. So it's a 27 kDa protein, and we have many publications on this discovery, as well as in terms of the different properties and the mechanism of action of P128. And okay, so I want to touch here on the modular organization. See, these lysins not only 128, all these phage lysins, be it ectolysin or endolysin. So they have a modular organization. So they have different domains which are having specific activity or specific functionality. So this gives a lot of window for protein engineering. Like you have the flexibility of swapping domains to look at new properties, and also having targeted mutagenesis to specific sections of each of the domain. To look for whether that could alter the specificity of the parent protein. So we also have had a lot of experience and success with respect to this. So we have actually done engineering with 128. We have had variants which are having a different specificity. So that actually is a big plus in terms of the engineering opportunities with respect to the license in general. So going next slide, I'll be talking about, okay, so P128, after we have identified P128, we went ahead with expressing. So P120 is expressed as a recombinant protein in E. coli expression system. And so we have actually started off from a shake flask experiment. So basically a complex media, shake flask culture, batch mode of fermentation. And from then on, we progressed to a fed batch fermentation. So we move on to all through the media optimization. We have gone to a fed batch mode of fermentation, and then to a fed batch mode of fermentation. So this has enabled us to increase the biomass as well as increase the yield of P128. So the typical purification overflow is something like this. If we start with the fermenter harvest and then go to that will be next step to the cell lysis. So it's an intracellular expression, and to release the protein, then we have a series of precipitation steps. We need to take care of the contaminant proteins and the impurity, and then we have two-step ion exchange chromatography for purifying the protein to homogeneity, and then ultrafiltration, diafiltration step mainly for buffer exchange or any concentration, sterile filtration, and then the storage. So at this stage, it's the pure protein and called as the drug substance in a regulatory terminology. So this is the purity profile of 128. We were able to purify it to homogeneity greater than 98% purity. So in terms of the expression yields, we have standardized fed batch conditions to get in the range of 7 grams per liter upstream, that is a fermenter expression. And then we have also optimized the purification methodology to handle higher biomass and get protein to great purity. So as one of the requirement when you are looking at protein or any lysine application for therapeutics in humans, one of the regulatory need in terms of chemistry and manufacturing controls is to have a purity criteria. So we need to have specifications in place to profile the purity of the protein, and we need to have a set of parameters, a different test, a battery of tests to actually say that each batch of protein is passing the criteria. So we have established the specifications for batch release for P128 and batch production method. Going next, I'll go to the properties, starting with the property. So one is we know that this is an anti-staphylococcal protein. So this data shows the MIC of P128 on MRSA. So MRSA being a super bug, that was where we started off with. So our MIC on, so this is a panel of 105 MRSA strains. So the left-hand side table, actually, the graph shows the MIC across these strains, and the right-hand side shows the MBC values on these strains. So MIC 90 of P128 across these strains was about 8 micrograms per ml. And this panel actually has it actually has a subset of several clinical isolates from different sources. Specifically, it has a set of 31 panel of global isolates, 31 isolates, which is representative of clinical isolates across the world. So we were able to see a good MIC with P128, and the MIC to MBC correlation was 2x. And also draw an attention to this point. Here, if you look at the comparison of MIC when we are doing MIC under in vitro conditions, when we check the MIC against CAMHB, the regular routine media what we use, and versus, because this is targeted for human application. So we were trying to see the MIC also in fetal cough serum and human serum. So one of the main point what we were able to see is this serum. In serum, actually, we get a very low MIC when compared to that in the media. So we were able to get the serum MIC was very low as against the 8 or this particular set of strains. It's around 4 micrograms per ml, and but when done in serum, it was ranging from 0.1 to 1 microgram per ml. So very low, about 4 to 64-fold low when compared to that what we see in the media. So this is actually a very, very important point going forward in terms of looking at clinical efficacy when we are going to administer this in humans. So that will enhance the potency of the molecule. So that is a very critical point. And then coming to the unique properties of 128. So these are some of the highlights of the. So this P120 is actually very highly specific to Staphylococcus species. So it is active both on RES as well as on coagulous negative staph. It is rapid in its kill, so it's going to kill in a few minutes upon contact because this being an enzymatic kill. And then it demonstrates strong synergy with standard of care antibiotics and across different classes of antibiotics. And it also has a very robust biofilm degrading activity. See, these are all very important qualities which you look for when you're trying to progress a molecule to clinic. And the last one is it needs to be active on the biological fluid condition. So I'll be touching on some of the data covering these in the further slides. So the first one is on the specificity. So this so here actually what we have done, this is an turbidity reduction assay or an ODFOL assay. So where we have taken 50 micrograms per ml of 128 and we have done exposed that to log phase cultures of both gram-negative and gram-positive and RES as well as other gram-negative pathogens. So we have taken Pseudomonas aeruginosa, Klebsiella, E. coli, then also S. carnosus and S. aureus. So what we see is P120 is very, very specific. So it was targeting only Staphylococcus, and it was not specific. It was not actually touching E. coli, Pseudomonas, or Klebsiella. So this is actually very good in terms of what we're looking for, because it will be very highly specific to staphylococcal infections, and the other one is it will also be narrow spectrum because of this, and hence it does not disturb the normal microbiome. So that is an important quality. And okay, the second one is actually how quick P128 acts. So this is a time kill assay, a TKK profile of P128 on SRS USA 300 and MRSA strain. Here you can see the, okay, so here what is being the y-axis is the CFU per ml, and the x-axis is the time duration. So here what we do is we take these cells and expose it to P128 daptomycin, vancomycin, all at 1x MIC for this particular strain. Start off with about 10^5 CFU per ml, and then periodically sample at different time points when we expose, take the cells, and this is a cell control with the drug of what we are testing. When we have them and sample and look for the CFU per ml over points of time. So what was striking is P120 was very, very quick in action. So we were able to see in about 30 minutes, we were able to get a complete kill by CFU enumeration. So this also says when we compare this with the standard of care antibiotics, the time record for kill is very, very quick. So it's very, this probably is due to the enzymatic nature of P128. So this is also a very, very important property. And the next one is, okay, so P128 synergizes across with antibiotics across all class. So we were, this is actually the P128 synergy experiment across different antibiotics, and this is actually the FSCI range. So clearly less than 0.5, indicating that it's a clear, very good synergy is being seen. So we were able to see P128 synergizes with different classes of antibiotics, and this is also an important factor. Looking at touching upon what BG had said. So instead of looking at a replacement for antibiotics, what we look here is also if there is an antibiotic. So antibiotics have been the workhorse and has been helping us over several years. And of course, the antimicrobial resistance is a problem which we need to tackle with. So if you look at lysins in combination with the traditional antibiotics, we are going to get an effective MIC because of the synergy aspects. So that is an important aspect. And then the other striking feature is actually the reversal of antibiotic resistance. So this set of data is actually, it is a synergy experiment, but the point is what we have taken is the antibiotic resistant strains. So we have taken MRSA here, and we have we have done a checkerboard assay for finding the synergy between P128 and oxacillin. So oxacillin is not expected to work, so we see a very high MIC. And P128, when we take alone works, and when we take a combination of that, it was very, very striking that it was a clear synergy. Less, the FAC index values were less than 0.5, indicating that there is a reversal of the antibiotic resistance when we have even the drug resistant pathogens. When you're taking the drug which is already resistant, with P128 we were able to see a clean reversal of the antibiotic resistance. So the same was observed across vancomycin as well. So the second table here is actually a panel of vancomycin resistant staph aureus. Again, vancomycin clearly showing a high MIC, but when in combination with 128, we were able to get a very good synergy. Same with strains done with linezolid resistant staph aureus. So basically, this is a graphical representation. So clearly saying that P128 synergizes with standard of care antibiotics, and the most striking is it synergizes with even the resistant drugs. So the resistant, making the antibiotic resistant, reversing it. So this we feel is also a very, very good aspect when we are trying to look at using P128 with an already existing standard of care treatment. And the next is actually a P128 action biofilms. So P128, so we have two different models here. It's a 24-well microreticulated model, and the other one is catheter model. So in both these cases, we have tested P128 at 8 micrograms per ml, and we have included, in this case, microdata, we have used daptomycin, vancomycin, linezolid at even 500 micrograms per ml. So these are antibiotic resistant strain has been used here, and we clearly see that P128 even, these are actually pre-formed biofilms. So biofilms are already formed, and then we expose it to the drugs at this concentration. So even from 2 hours onwards, we were exposure to the drug at 2 hours, 4 hours, 24 hours was the readout. And this is crystal violet staining post these time points. So we clearly see that P128 even at 8 microgram per ml was able to give complete eradication of biofilms. So which is a very, very positive aspect looking at the therapeutic angle. And also this is actually the catheter biofilm model. So this is actually the uninfected catheter. So here we do a safranin staining post the biofilm formation, as well as post the treatment with the drug. So here also we are not able to see any culture here, biofilm here when we have treated with P128 at 8 micrograms per ml. So we also did the electron microscopy of these catheters. So this is the cell control where we are able to see the bugs there live and thriving, and the other one is actually the P128 treated conditions where we don't see the bugs. So clearly biofilm is a problem, and P128 eradicated biofilm had a very good biofilm eradication property. And then I'll go to a few experiments which mainly to say whatever we are seeing in vitro has to translate in vivo. So this experiment is actually an experiment where we have done a dose ranging study with 128. This is an IP model, so intraperitoneal challenge with but this is done in female BALB/c mice 8 to 9 weeks old, and we have used SRS call MRSA for this. And the intraperitoneal challenge is about 10 power 9 CFU per ml, and treatment was done post 3 hours of challenge with P128 at 0.21 and 5 mg per kg. And survival was monitored for 72 hours. So this is the graphical, the representation of the results. So as you can see, this infection control, the survival is one below zero, close to 0%. There are no survivors at about less than 24 hours. But P128 at 0.2 mg per kg was giving around 20% survival. At 1 mg per kg, around 40% survival. And when we were using a 5 mg per kg, clearly giving around 65 to 70% survival. So this clearly indicated that P128, the animals treated with P128 showed a dose dependent survival. And then also have an experiment showing the synergy. How does the synergy what we see in vitro translates in vivo? So here, this is also a similar experiment where we have done with we have challenged. This is also an IP model where we have used BALB/c mice 8 to 9 weeks old. We have challenged with SRS call 10 power 9 CFU per animal, and then post 2 hours we have done treatment with either P128 at 0.2 mg per kg or vancomycin or both. Vancomycin, it's two times. It was administered 2 times as per the standard procedure. So 12 hours after the first dose, vancomycin second dose was also provided. And again, here we were looking at survival at 72 hours. So looking at the graph, we see that the infection control was about 12%, and one survival was only 12% after 72 hours. So the infection is established. And P128 alone gave a 30%. So we have used a sub-efficacious dose to appreciate the synergy. So we have used 0.2 mg per kg, which was giving about 30% survival. And vancomycin alone gave about 46%. And when we use both in combination, we were able to get about 84% survival. So clearly, whatever synergy we were able to appreciate in vitro, in MIC, the checkerboards, we were able to see that they were translating into the in vivo studies. So actually, we have done a lot of toxicology studies post this. So I'm not touching upon that in this presentation. So we have done we have done dermal toxicology study in rabbits, and we have also done IV infusion study with toxicology study with mini pig. So in all the doses what we have tried, we have seen it was P120 was very well tolerated and did not have any toxicology, was not toxic, and it did not have any adverse effect. So with that, I'll go to summarize the presentation today. So antimicrobial resistance is a problem, and it has thrown up challenges for drug developers. And ectolysins are a very good example of direct-acting non-traditional antibiotic. So P128 being an ectolysin showed very good, very potent in vitro and in vivo efficacy against SREs. It not only killed P128, not only killed SREs on its own, but it was able to effectively synergize with standard of care antibiotics, not only on the sensitive ones, but also on the drug resistant ones, thereby giving superior endpoints in terms of reversion of antibiotic resistance. The stage where we are right now, we are progressing P128 for systemic use in bacteremia with infective enterocolitis. So we have had a pre-IND meeting with US FDA, which is completed, and we are working towards forwarding the molecule to clinics. Before I wrap up, I'll go to the next slide here. So this is basically on where we are using the same platform technology, what we have used for discovering P128. We have been using this for several other bugs as well.
Sukumar, we can't hear you. Have you lost the connection briefly?
Sukumar, we can't hear you. Okay, so shall I just finish up, Urmi?
Yes, please. Yes, please.
Their meeting domain.
Sukumar, so I asked you for the last 1 minute. Can you repeat?
The gram negative? You mean the gram negative?
No, even after the gram positive.
Sorry, Viji?
Start from the gram positive bacteria.
Okay, so we have used this platform technology for identifying ectolysins which are active against other gram positive bacteria as well as gram negative bacteria. So we have running programs on this. And we have also used these bioinformatic platforms to identify novel bacteriocins. So bacteriocins are also have domain architecture. So they are also modular. So we have been able to use the same platform to even identify novel bacteriocins, which are targeted towards escape pathogens. So, so this is where we are with respect to the other license using the same platform technology. And with that, I would come to the last slide. So this is the Gangajan team, and they were responsible for all the the work which I presented right now. And thank you. And that's it I have.
Thank you so much. BG, you want to wrap up something, or should we shoot the questions?
No, I'm fine. We'll shoot the questions.
Okay, so first question is a generic one. What challenges did the team face while developing this license as therapeutics? Any specific challenge you want to bring your attention to?
Yeah, clearly, this is anticipated. Lysine is a large molecule. It's a protein. So the first reaction that you would get is, oh no, this will be an immunological disaster. But that's not true. Our experience, in fact, We have injected P128 IV. We have done pilot talks in mini pigs. There's a minimal immune response. This has not been a problem. There is no hypersensitivity. So whether this is unique for P128, I don't know. But clearly taking proteins forward as therapeutics, you've got to remember this as something you have to deal with. And second is, of course, you must remember that the distribution of a large molecule is something not completely understood so far. So something we obviously have to work out by trial and error. That doesn't mean that these are hurdles. All I'm talking about is small molecules. There are challenges, and there's so many advantages for license, so you can deal with some challenges.
Yeah, it's true. So one question by Aditi Singh, you have already replied, but I may repeat for everyone. As she is asking is, she had a question if P128 converts only the resistant Staph aureus. So BG has already answered that P128 is equally effective against weather susceptible Staph aureus or the ones which are resistant to methyl, resistant to the antibiotics. So that's the beauty of license. They actually can kill both kinds of bacterial strains.
Absolutely. And the only thing I want to add, which probably we should have mentioned, is that when we reverse, we have mentioned, but it sort of slipped, is that when we talk about reversal, we are using quite very low concentrations of the lysine, which allows reversal.
Yeah, that's good. So that you don't have to use a large concentration of the enzyme, but then it does the job of making the bacteria susceptible.
Exactly.
So one more question is from Ajit Madhwan. He asked, the bacteria is, he wants to ask, out of the two, bacteria is likely to develop resistance against lysins or the complete phage?
Yeah, so it's a good question. Phage lysin, phages are generally -4, -5 is the resistance rate. While we found that with P128, and it has also been found with other licenses, which I mentioned earlier on, our resistance rate are in the order of about -8. So it's not a really worry to deal with. And secondly, remember, we are positioning it in combination. So where we don't find any mutants at all.
Right.
Okay, so to add to this point, with respect to phages, resistance of phages versus the lysine. And phage, there are several other factors. We are talking about the receptor availability, so that also plays a role, but that is not the case with lysins.
Right.
So, and also the phase of the growth. All these are, if you try to compare contrast between phages and the lysins. So, receptor or the presence of superinfection immunity. If there is a lysogen sitting in having the same similar like the phage outside, the phage is not going to cause and not going to give a clearance. But those are all points which are clearly an advantage for the lysins. So lysins act on those. Suppose if phage K, for example, if you take phage K, even if that is resistant, if it is not able to kill a particular strain because the non-availability of a receptor, that would not be the case for a lysine. The lysine is still an enzymatic; it will still do the killing. So those are some marking advantage, and the presence of a lysogen or a presence of a similar phage inside or not will be playing a role for phage, but not for lysins. So that also, in terms of the resistance angle, that plays a critical role.
Okay. So this is a question about what is the public acceptance? I mean, though you are in the clinical trial phase, but do you anticipate acceptance of, I mean, given that we might have other protein antimicrobials also. So what is the probability of public acceptance of this novel therapeutic agent is the question.
So when you say public, are we including clinicians into that? That's a so the point is clinicians would go clearly by safety and efficacy, both of which if you demonstrate, most clinicians are happy to use it. So we've not found any clear, any dogma saying we will use only small molecules. As far as the general public is concerned, remember, we are much more primed today because we've got many protein therapeutics out there. And most people have heard about it. So I don't see this as a problem. And I think it's mostly driven by the acceptance in the clinical community.
Right. The next question is, so we'll take questions for another 5 minutes. And I would request Mr. Sukumar, if some of the questions which could be possibly answered in monosyllables or in short answers, you could take those questions in the chat box. I'm taking more which will interest a large community because people are not so well aware of the potential of license. So somebody wants to know, how long does it take to finish the study from phage isolation to purifying the protein and then testing them on biofilms?
I can only tell you how long we took, because remember ours. So one of the things that you must remember is that ours is not a straightforward natural protein that we took from the phage. We optimized both the binding, especially the binding. In fact, P128 has got 2 binding sites for the peptidoglycan. So we took us about 4 to 5 years to bring it to early clinical stage.
Okay, so guys, you
So
need
if
to.
I did it again, I'd probably do it in 2 years, but I don't know.
Yeah. And this is a specific question. How did you inject P128 in mice? Was it intraperitoneal? And did you check the serum IgG, IgM levels after injection? Was there an immune response in the mice?
Right. So in the animal infection studies, the Sukumar is okay, I take it, right?
Yeah, sure.
We have done both IV, IP treatment, and there's lots of data on that. There are 2 parts to it. One is, have we seen any acute hypersensitive reactions? No, we've never seen any of them. Second is, have you seen development of antibiotics? We've desperately tried to develop antibodies against P128, and we've in mice and rats and rabbits have not been very successful. So on the other hand, when we did mini pigs, where we have done 14-day dosing, we have seen low antibody responses, but those responses were not neutralizing, which is the most important aspect of that study.
Okay. Ranjan Gupta wants to ask, do phage product development follows the same regulatory pathway by DCGI in India as for the other drugs?
Urmi, I can't answer the DCGI India, but FDA follows the same as for any other antibacterial. Of course, from a safety perspective, they would focus on immunotox, which is not generally a problem with small molecules, but this is something they would do for a large molecule. But on the other hand, small molecules, they do a lot of studies on excretion and other metabolic modifications, which is not the case with the protein molecule. I would expect that DCGI India follows the same pattern.
Dr. Sukumar wants to ask, have you seen emergence of resistance against P128? I mean, he has heard somewhere before that there is a development of resistance. So have you identified genes conferring resistance to P128?
We have done experiments where we are subjecting to on a plate on the in vitro experiment to look for resistors, and we were able to isolate resistors. And they were mainly when we tried to understand what is a mutation, it was mostly on the FEM gene. So if you look at this, is actually the resistance is linked to the mechanism of action of 128. So action 128 actually works on the P128's mechanism of action is it actually targets the pentaglycine bridges in the side chain. So whenever there is a modification there, that is what it would lead to a resistance. So whatever resistant mutants we were able to get at the lower frequency, when we were trying to sequence and find, those were actually mutations in the FEM gene, which are actually responsible in the pentaglycine bridge formation.
This is an interesting question from Swati. She wants to know, can we purchase P128 from Gangajan? So you have a potential buyer already, listening to your 40-minute talk.
If it is for research purposes, we are happy to share, but it's not commercially available from that perspective.
Sure. And she's saying, do you have more such license with broad spectrum activity? Are you working on any of those?
Sorry, come again, Urmi?
Do you have a license with broad spectrum activity? Like this one, because it's a fusion one, so it's very specific to Staph aureus, but are you working on license with broad spectrum activity?
Most of the license that we have studied are with narrow spectrum, and our sort of USP has also been keep it as narrow as possible, because we believe that's important from a microbiome perspective.
So I'll take a break. I want to ask Joe, are you there? Has Joseph Campbell joined? I don't know. Oh, hi, Joe. So Joe is here. So now we cannot push the time. And, I want to thank VG and Sukumar profusely. If you look at the number of questions which are here on the chat box, and let me tell you, my students are also preparing a Google Doc where they are picking questions from the YouTube, because we have 50 plus participants there as well. But you know that given the time constraints, we cannot ask all the questions, though I was just loving this question answer session. And it's, it tells a lot about how interesting the talk has been for everybody, that, so many questions are there. But in an interest of time, it will not be possible. Sorry, guys, it will not be possible to take such interesting questions. So we will try to communicate these questions to Dr. PG and Mr. Sukumar. If they are generous with time, they might be able to reply to those questions. We have to move with our next speaker. So thank you.
We'll be happy to answer those questions.
Yeah, I will definitely send your way. And these are really fantastic questions. So, but then, yeah.
And you've also been working on license. You can answer some of them.
I can answer the generic ones, but not specific to P128. Yes, I can definitely to those which are general ones, but like what's the stability of P128, whether it is broad spectrum, whether I can buy. These are the questions I think I will send to you, and the general one I will take. Yeah, that's a good option.
No problem. I will do that. Thank you very much.
Thank you. So thank you so much, Dr. BG. I mean, indeed a great pleasure. We have been wanting to have Gangajan on IBRC for a very long time, and you are very kind enough to accept our invitation and give such a detailed account of P128, which is so much talked about. So thank you so much. And Ranjan Gupta is invisible, but he has been the person behind organizing this. So thank you, Ranjan, and Joe. So Joseph Campbell is already here. Thank you, Dr. Joe, for joining. Now I will quickly introduce you all to Dr. Joe, and then Dr. Joe will introduce to Professor Fisherty, who has also already joined. Thank you so much, both of you. I'll take just 2 minutes because Dr. Joe is going to tell us about the landscape of bacteriophage research in the United States. And he's the Research Resource Project Officer in the National Institute of Allergies and Infectious Diseases, that is NIH, in NIH. He's responsible for several tasks there, but I would draw your attention to one of them, which is he runs the Division of Microbiology and Infectious Disease, which we call DMIT. So he runs DMIT's Bacteriophage Interest Group. And the goal of this group is to work with the FDA and other federal agencies to promote the clinical use of bacteriophages. So thank you so much, Dr. Joe, for joining. And so you may please tell us about the brief overview of the landscape of bacteriophage research in the United States, and then kindly introduce Professor Fisherty to our audience.
Wonderful. Thank you so much for having me. Can everyone hear me?
Yep.
Yeah, we can hear you.
Great. Thanks, and thanks for making the time more convenient for us than perhaps for you. But it was great to hear the first talk, and I'm sure Vince, as usual, will give a wonderful second talk. I'm going to share my screen and present a few brief slides to make a few remarks about the NIH and how we are trying to promote bacteriophages and license. So let me see if I can. Can people see my screen now?
Not yet. You'll have to share it.
Can they see it now?
No.
All right. I'm going to send these slides to you, but I'll start talking.
Maybe you can read out from the screen from your slide.
Yeah, I'll do that, but I'm just trying to see if I can share it. I'm going to share it with you as an email, and then if you have a chance, you can share them, but if not, I'll just go through them. So the NIH has an interest in bacteriophages, and one of the things on the second slide, what I have is the NIH in 2019 released a document outlining its antibiotic research framework. And there's a link for it there, which I can send people. Of note for this is that non-antibiotic approaches to combat antimicrobial resistance were one of the promising areas highlighted in 2019. And of course, this includes phages and lysins. And we support phages and lysins in 3 ways. We give our own grants, and we have what we call other funding sources that we support indirectly. And then we have what we call our preclinical services. And the next slide, I had a couple links where you can look at our grant opportunities, but if anyone's interested, you can contact me about that. And then I was going to highlight a couple other funding sources. The Antimicrobial Resistance Leadership Group, or ARLG, is something you might want to look at. They provide clinical trial support. They provide clinical isolates and industry consultation, and we fund them. And I would encourage you, if you're interested in trying to develop your products, to Google them and you look them up. The other is the CARB-X, the Combating Antibiotic Resistance Bacteria Accelerator, which is funded by NIAID and mostly by BARDA in the US. And then I should say broadly, and I was really pleased with the first talk because, and the question about the acceptance of these products, I think the answer really nailed it on the head. I mean, I think if clinical data is there, and that's what we need with phages and with lysins, I think the acceptance will be there. I mean, certainly phages specifically have gotten a lot of publicity recently because of some of the high-profile compassionate use cases where there's been apparent success, but of course, it's been impossible to really see whether there was success because there weren't placebo controls, and some of the trials that are happening with both phages and lysins are really encouraging. And I think the community, the clinicians there's a lot of them are eager to use it, but there are many who are still skeptical because as of yet, there haven't been clear, compelling clinical data. But fortunately, as I outlined in the first talk, and I'm sure as Vince will outline in the second talk, that's not for now. People are really trying to push for that. And I will say that we have a variety of preclinical services. We have a biopharmaceutical product development series, an in vitro assessment for antimicrobial activity, and preclinical models of infectious diseases, all of which are intended to be gap filling to help you develop your product. And you can reach out to me via Ume to learn more information about those. And I put links on the slides, and I can share the slides later. But now let me get to introducing, stop the share now. Now I figured out how to do the share. Now I want to introduce Vince. So it's a great pleasure. We had a great first talk, and I'm sure we'll have a great second talk. Vince Fischetti has been at the Rockefeller for a very long time and has been working on Lysins for a considerable amount of time, and has, in my mind, been one of the true leaders in the development of this technology for the treatment of infections. And so it's a great pleasure to introduce Vince, and I will let Vince take over now.
Thank you, Joe. I appreciate it. And thanks, Hermie, for the opportunity to speak to everyone here. Let's see if I can share my screen. Can you see that?
Yes, please. Yes, we can see.
Great. Okay. Let me just minimize this. Okay. So can everyone see everything okay?
Yes, perfect. It's perfect. The slide, the first slide we can see, it's all good.
I just can't get rid of the side. Pointer won't let me do that. All right, I'll do my best to get around it. So today I'd like to just give you all around understanding of what Lysins are. And there may be people that are listening that are really not up to what Lysins are. They may be new in the field, and I'll try to give you an outline of that, and then get into some of the things that we're doing in the lab and where we think Lysins may be going in the future. The important thing here is that resistance is a major problem. Antibiotic resistance is a major problem. And when the escape pathogens was, that acronym was mentioned about 10 years ago, those organisms were resistant to antibiotics. And that was the reason for this escape pathogens being listed. But now it's 10 years later, and these organisms are still resistant, in fact, multiply resistant, even more than they were 10 years ago. So it's a more serious problem that we're dealing with at this point. And we must deal with alternatives to antibiotics. And we've taken oops. Huh. I don't know if my, oh, there we go. We developed Phage Lysins as an alternative to conventional antibiotics in hopes to get around this resistance issue. My slides are not moving forward. Let me get out for a minute and see if I can, what's going on. The pointer, I think, is causing an issue. That's the issue. Can't advance my slides. There we go. So, okay. So, phage lysins are enzymes used by phage to get out of the infected cell. Every 48 hours, half the bacteria on Earth are killed by bacteriophage through the lytic cycle, which makes phage lysins the most effective bactericidal agent on the planet. These enzymes are killing bacteria constantly, and they've evolved for this purpose. So they are extremely effective enzymes that have been developed over 1 billion years. And since lysins appear to be essential for phage survival, they have evolved to avoid resistance. And so resistance to lysins is yet to be observed. No one's found resistance to these enzymes. So I don't think we have the same problem as antibiotic resistance, in which bacteria are bathed in antibiotics from the time for millions of years. So they've developed systems to get around antibiotics. But since lysins are essential for the survival of the phage, we really haven't seen resistance. It may take hundreds of years for resistance to occur in the same way that they've occurred against antibiotics. So just to put everyone on the same page, I'm sure everyone really knows about lysins, but just so there are people out there that don't understand what lysins are. This is a gram-positive organism. There's a cell wall. Here is the cytoplasmic membrane. A bacteriophage are viruses that interact specifically to receptors on the cell wall of the bacteria. Once those interactions occur, the phage inject their DNA into the host cell. Once that DNA gets into the host cell, it forces the cell to produce more phage. And once those phage are assembled, they have a problem. They need to get out of this infected organism. They produce a lysin that gets through the membrane through the production of a phage-encoded holin. And once that enzyme hits the cell wall, it cleaves the bonds in the wall. And since the pressure inside the bacteria is greater than the external environment, the membrane externalizes, releases the phage into the environment. This cycle is phage therapy. And that's one way of killing bacteria. But what we've done is purify the lysins. All our lysins are now made recombinantly. And if you take the recombinantly produced purified enzyme and add it externally to a gram-positive or a gram-negative organism, it will do precisely what it did from the inside, cuts a hole in the cell wall, membrane externalizes, explodes, killing the organism virtually instantly. So to give you an idea of what this looks like, here's a Bacillus anthracis. You can see this organism is completely intact. But after about a minute of treatment with lysin, you can see that the cell wall has collapsed, the cytoplasm has been released, and the organism has been killed. And you can see here, there are a couple of organisms that have not been hit yet, but the bacilli on either side have collapsed. And you can, with the thin section electron micrograph, you can see the membrane externalizing here just before the cytoplasm is released. So it's a very rapid, very efficient way of killing the organism. It's basically taking a balloon and punching a hole in the balloon. The whole balloon collapses and you're left with the balloon afterwards. If you add too much lysin, you could cut up the cell wall into several fragments, and those small fragments could be inflammatory. But the therapeutic doses of lysin that are being used are sufficient to punch a hole in the organism, but not to cut up the organism in lots of little pieces. I'll just show you in a video basically the same occurrence, but this is a little more dramatic. And I'm sure several of you have seen this before. Now, just remember that gram-positive organisms are about 15 to 25 atmospheres of pressure inside that organism. So once you punch a hole in that organism, they will eject their cytoplasm and they will explode. And this is a video using phase contrast microscopy. So what you see is the cytoplasm in dark. Once the cytoplasm is ejected, basically the organism appears to disappear. So we'll start the video and you can see what happens here. If you follow this organism here, you can see the membrane externalizing organism disappears because you don't see the cytoplasm. If you follow any organism, it will eventually disappear. And this is very close to real time. So if you add more enzyme, you see more destruction. Less enzyme, it takes a little bit longer. So basically, these organisms look like this. They have a catalytic domain at one end and a binding domain at the other end. This is a gram-positive enzyme. They're anywhere between 25 and 40 kD in size. And these two domains are held together by a flexible linker. And the catalytic domain is either a muramidase, a glucosaminidase, endopeptidase, or L-alanine amidase, which are the major bonds in the peptidoglycan. At the other end, you have the binding domain, which binds at high affinity to an essential cell wall associated substrate which is usually a carbohydrate in the cell wall of the bacterium. So once the binding domain binds to its substrate in the cell wall, it positions the catalytic domain to cleave the bond adjacent to the binding site, thus killing the organism. And the gram-negative enzymes, they don't have binding domains. They're essentially catalytic domains and generally lysozymes with a charge region at one end of the molecule. In most cases, we see them at the C-terminal end, but you will see them at the N-terminus as well. But the great majority have these charge regions, positively charged region at the C-terminal end. So where do we get some of our lysins? So initially we collected phage from the environment, either water, soil, etc. Removed the bacteria by filtration, collected the phage, extracted the DNA by shotgun cloning, and identified lytic activity against the target bacteria. And you can see here, this is a clone. This is the lawn of the target bacteria, and you can see this halo around this clone that secreted the lysin to kill the target organism. This is very tedious. It takes a long time to do, and you're limited to the concentration of the types of phage that you have in the population. We then decided to induce phage from several strains of the target species. That way you could get phage or lysins that are directed against the target species. In that case, we extract the DNA from those phage and then identify the lytic activity against the target bacteria in a similar way. In this case, we could identify lysins more specific for the target species, where it is in the environmental case, it's a hit and miss whether the phage is going to be there or not. But now we have more control over it, and we're identifying lysin classes from protein sequence database, and we developed an algorithm to identify the most effective lysins using an algorithm we developed in the laboratory. And just to give you an example, we call it this algorithm a LIAR algorithm for lysin automatic research. What we've done here in this example, we've identified lysins against E. coli with about 14,000 genomes in the database with 75 million translated coding sequences. And using the algorithm to filter the sequences, and then with some a manual analysis looking for lysozyme domains, if we're looking for gram-negative enzymes, particularly against E. coli. In the end, we end up with 10 clusters, and we identify 10 enzymes out of those clusters. And you can see in the next slide, you could see these various clusters. We've identified, we take one enzyme out of each one of these clusters, have them synthesized and expressed, and we look for activity. If we find a cluster that has a high activity, we'll go back to the other lysins in that cluster and sequence more of them, because it looks like those would be the best ones to identify. So we've been doing this for the past couple of years. We've been very successful in identifying enzymes much more efficiently, much more quickly. We can identify new lysins in a matter of months, whereas in the other ways, it takes several months in order to be successful. So using these various methods, we've successfully identified lysins from a number of gram-positive organisms, basically all the major gram-positive pathogens. And the past few years, we've been looking at the gram-negative organisms as a gram-negative lysins as well. And we've covered in this series all the escape pathogens, the ones that are causing the major problems with resistance. And we've tested them in a number of animal models, either those developed in our own laboratory, or we've collaborated with people with unique animal models, and we've sent them lysin to see whether it would work in their systems. And you can see over the years, you can look at our publications, we've published a number of papers on all of these animal models to show that these enzymes will work under these conditions. Originally, when we discovered lysins as a therapeutic, we thought we could only use them topically in most instances and very limited ways in vivo, but we found that gram-positive lysins may be used nearly in all applications where antibiotics are used. So if you can use an antibiotic to control an infection, you can also use a lysin to control that same infection. They can get into the tissues and kill the organism within that infected organism, infected animal. So let me give you a couple of examples of the models that we've used over the years. Here's the MRSA bacteremia model using PLISS2 lysin. Here's a mouse where you give 1/10⁶ MRSA intravenously. 3 hours later, if you take some of the animals and you look at the organs, all the organs are infected with MRSA. We then treat intraperitoneally with 1 milligram of lysin and then measure survival. This is a very straightforward experiment. You can see here, the animals treated with lysin, over 90% of those animals are protected from infection, and those animals treated with just buffer, most of those animals die very quickly. Basically what we're doing here is taking the bacterial load that's in the animal, reducing it by several logs because of the rapid action of the lysin, and when we reduce the bacterial load by several logs, the animal's host immune system can clear up the remaining organisms. I don't believe in all of these experiments that we're killing every single organism. We're just reducing them very rapidly so that the host immune system can clear the infection. I think there's a real good use for these lysins is secondary bacterial infections. Secondary bacterial infections are a result of upper respiratory viral infections. The organisms responsible are Strep pneumoniae, Staph aureus, and Strep pyogenes. And these are organisms that colonize the upper respiratory tract in the nose and the throat. For example, influenza, up to 25% of patients that get influenza develop a secondary infection. And in most cases, in all of these secondary infections, they're usually pneumonia. SARS, several years ago, there was SARS outbreak. They found that 25% of the cases had secondary bacterial infections, and that was published in one study. And there are many studies now. I'm just citing a couple of early studies on COVID-19. Co-infections occurred in 15% of all cases and 50% of non-survivors. And 93% of critically ill patients receive antibiotics due to secondary infections. So in all, it's about a third of the COVID-19 patients develop secondary bacterial infections. And we feel that we can use lysins could be used to decolonize the upper respiratory tract early on during infection to reduce the pathogens, these organisms, Strep pneumoniae, Staph aureus, and Strep pyogenes, from the upper respiratory tract to reduce secondary bacterial infections. This is a model of secondary infection. It's otitis media. It's ear infections. Children and adults have otitis media, Staph aureus, Strep pneumoniae in our nose. When a child gets an upper respiratory viral infection, those organisms will migrate to the ear and cause ear infection. In this model, we take luciferase tags Strep pneumoniae, and these are delivered nasally to the mice. And as you can see, once they're colonized, you can see the fluorescence in the nose, bioluminescence in the nose of these animals. We wait 7 days and treat with buffer. And then 4 hours later, we give these animals influenza. And you can see 72 hours after the influenza, the organisms are now migrated to the ear and causing ear infection. If we do the same experiment, take the luciferase tags Strep pneumoniae, colonize the mice, and 7 days later, you can see they're colonizing the nose of these animals. We then treat with CPL1 lysin, wait 4 hours, and then treat again with lysin, and then treat with influenza. You can see that after the 72 hours, there are no organisms in the nose of these animals, and therefore, if there's no organisms in the nose because they've been removed by the lysin, there's no organisms that move to the ear, to the middle ear. So this happens in 80% of the time we can cause this infection, and 100% of the time we can remove them from the nose and prevent the secondary infection. So we believe that we can use lysins to decolonize the upper respiratory tract of people early on in their infection to prevent or reduce the amount of secondary bacterial infections. The one advantage of lysins that they destroy the biofilm. And the reason for this is that the bacteria are part of the biofilm. The biofilm is formed with secretions from the bacteria. They're composed of DNA, polysaccharides, and proteins that are secreted in this matrix, and the bacteria are part of the matrix. And if you take a lysin and destroy the bacteria, since they're part of the matrix, start to fall apart, and it causes the biofilm to collapse. And you can see here in this experiment where we use lysin to destroy the biofilm on these plastic dishes. If you take the plastic dish, you form 24-hour biofilms, in this case with Staph aureus. And then we can treat either with daptomycin at 1,000 MIC, or vancomycin at 1,000 MIC, or linezolid at 1,000 MIC. And as you can see, that there's no effect over several hours of treatment with these antibiotics. But 1 MIC of lysin, within 2 hours, you've destroyed the biofilm. And you can see here in this confocal microscopy, within an hour, you can see it very clearly that the biofilm has been destroyed. So basically, you're taking it and destroying it from the top down. You're just removing the organisms and killing them as they're disrupted. The biofilm falls apart. This is just a model of pneumococcal endocarditis, which is very difficult to treat. And it is a biofilmic infection where you get vegetations on the heart valve of the bacterium. So in this case, we're treating an endocarditis with lysin against pneumococci. So the way this experiment is performed is we take a catheter, run it down the jugular vein of the rat, cause damage to the heart valve, and then add 10 to the 7th penicillin-resistant pneumococci 2 days prior to the challenge. We then challenge with pneumococci, and then we treat with 10 milligrams per kilogram bolus of lysin, and then a continuous IV infusion of 5 milligrams per kilogram per hour for 6 hours. And if you look at the buffer-treated animals in blood, the amount of organisms in the blood has remained very high, about 10 to the 5th. And if you look at the aortic vegetations, the animals are sacrificed, the aortas are removed and ground up, and you can see that the bacterial counts over 2 hours have gone up in these control animals. However, the ones treated with lysin in blood, the bacteria dropped significantly, and even on the heart valves within 6 hours, there's a significant drop in the amount of bacteria on the heart valves. This is extremely significant because you wouldn't see that in humans. It takes days of IV treatment to see any effect of the destruction of the biofilm on the heart valve. So it's a very efficient way of killing these organisms on the heart valves. We're now working on, we have a paper coming out soon regarding wound infection. We think lysins could be used to prevent wound infections in hospital situation. And we looked at orthopedic and cardiothoracic surgeries. About 2 million of those are done each year with about 1 to 2% infection rate, a fairly high infection rate considering. If you look at surgery for malignant pelvic tumors, they have about a 30% infection rate. This is a rate that is one of the best hospitals in the city, and that's Memorial Sloan Kettering Cancer Center, and they have a 30% infection rate for malignant pelvic tumors. These are surgeries where it takes hours to produce produce these surgeries, and they have a high infection rate. And military wound injuries are most common, are Staph aureus. So we developed a model, a rat wound model, a MRSA infection, where we make an incision in the back between the scapula of a rat. We then treat with either 10 to the 5th or 10 to the 6th MRSA in the presence of glass beads. You need glass beads because you need some foreign body in the wound in order to allow the organism to become more virulent. If we didn't add glass beads, we'd have to add 10 to the 9th organisms to cause the abscess. And in normal situations, a titanium implant or whatever would be the foreign body in that say a knee replacement or a hip replacement. So we then add glass beads in this model and the bacteria. The lysin is then added sometime afterwards, and then the wounds are closed. And then about 5 days later, we open the wounds and check for the extent of infection. So in this experiment, we call a high dose, low dose experiment, where we try to simulate real world contamination with the low dose, about 10 to the 4th. It's the lowest we can go. So the rats are surgically opened, infected with either the 10th to the 7th or the 10th to the 4th MRSA. Then after 10 minutes, the wounds are then treated with lysin and with the buffer or buffer as a control. And then they're surgically closed. And 5 days later, the animals are euthanized. The wounds are then opened, swabbed, and tissue samples are taken and determine the CFUs per gram of tissue. So in the high dose experiment, you can see that the animals that were treated with buffer, you can recover about 10th of the 7th, 10th of the 8th organisms. And if you look at the wound, they look terrible. They're a large abscess. They don't heal properly. They're pretty nasty. Whereas the animals that were treated with lysin, with the high dose of bacteria, most of those organisms were below detectable limit, and there were only three animals that had positive counts, and those were very low counts. And you can see in the backs of these animals, they're basically healed. The low dose treatment, you can see a wide range of recovery in the buffer treated organisms because we're adding so few organisms. But all of the animals that were treated with lysin were below detectable limit. So in all, 19 of the 22 animals in this whole experiment treated with lysin in both dosing groups had 0 colony-forming units. So it was very efficient in removing the organisms from the infection site. So we believe that lysins could be a more effective irrigant than saline during surgery. So we believe that after a surgery, if the surgeons use lysin to irrigate the site before suturing up the patient, we could reduce the amount of surgical wound infections. So let's talk a little bit about gram-negative lysins, which we've been working on the past few years. We worked with Klebsiella, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter strains, and we've been testing them against a number of animal models. These enzymes, as I mentioned earlier, look different. They're only catalytic domains, mostly lysozymes, and they have a charge region, a positively charged region at their C-terminal end. And this charge region, in some cases, resemble antimicrobial peptides. In some, if you do modeling, they appear like helix-turn helixes. We can use the whole enzyme to protect against infection, but we can also take this peptide alone or modified in some way to prevent infection or to cure an infection. Then I'll show you various examples of this. So let's look at how gram-negative enzymes work naturally. So you have the cytoplasm, the phage has infected this organism, has replicated it. Now the lysin is now trying to get through the cytoplasmic membrane, but it can't until a holin is produced. So this lysin, as I described here, in green is the catalytic domain, and red is this charge region. So once the holin is produced by the phage encoded holin, the lysin can go through the hole, interacts with the peptidoglycan, and the catalytic region will cleave the peptidoglycan. Now, since the pressure inside a gram-negative is much less than the gram-positive organism, it's only about 2 to 3 atmospheres, the outer membrane may keep the phage from getting out because the pressure is not enough to disrupt the outer membrane. So the lysin has this charge region which attempts to disrupt the outer membrane. And once the outer membrane is disrupted, then the phage are able to get through and out of the organism. So it's because of the pressure differential, the small amount of pressure on the gram-negatives, that the charge region is used to help disrupt the outer membrane. If you used a whole enzyme from the outside, it does precisely what it does from the inside in reverse. The charge region is used to, will disrupt the outer membrane. And once it disrupts the outer membrane, the lysine can now hit the peptidoglycan. And now the catalytic domain can cleave the peptidoglycan. And now the charge region is still available to disrupt the inner membrane, and that kills the organism. And if we look at this electron micrograph, 15 seconds after treatment of this Acinetobacter, you can see all you see is bacterial debris because the whole organism has been destroyed. The outer membrane has been disrupted with the charge region, and the peptidoglycan has been destroyed with the catalytic region. And you can see a couple of organisms still remaining here because it's only 15 seconds after treatment, and they will be destroyed very quickly. The problem with this method is that it produces a lot of endotoxin because you're destroying the organism completely. Now, just taking the peptide alone, and this is the isolated peptide, now add that from the outside, what it does is what it's designed to do is disrupt the outer membrane. And in doing so, it allows it to penetrate into the periplasm, interacting with the peptidoglycan. But since there's no catalytic domain here, and it's small enough to pass through the peptidoglycan and disrupts the inner membrane, killing the organism. And you can see here in this electron micrograph, after 2 hours of treatment, all you see a bacterial ghost, pretty much empty bacterial ghost, in which the cytoplasm has been ejected and killing the organism. In this case, despite the fact that you still get some disruption of the outer membrane, there's still endotoxin, but there's much less under these conditions than in the whole lysin conditions. And just to give you a differential of activity between the peptide and the whole enzyme, this is lysin against the Acinetobacter, and this is a number of strains of Acinetobacter. You can see about the controls, about 10 to the 6th organisms in these controls after treatment with the whole whole lysin in green. You get about a 3 to 4 log kill, which is quite reasonable. But after you treating just with the peptide alone, you get a 6 log plus kill. So the peptide efficiency of peptide killing is much better than the whole enzyme. The problem with these gram-negative lysins is the charge region, we believe, becomes activated when it interacts with serum proteins because of the, we think it becomes interacting with the serum proteins and gets pulled out of solution. And you can see that here in this experiment with 2 lysins against Pseudomonas aeruginosa, PA03 and PA91, increasing amount of human serum. And by the time it gets to 8% serum, the enzymes are inactivated. They don't work. The good news is that they will work in the presence of lung surfactants. Survanta is a lung surfactant, and these enzymes will work in lung surfactants. So despite the fact that we can't use them in serum, we can use these enzymes topically in skin and in lungs. And I'll give you an example of that in the lung model of Pseudomonas infection. Here we take a mouse, treat them with Pseudomonas aeruginosa nasally with 50 microliters of 10 to the 8th organisms. And then 3 hours and 6 hours later, we treat with 90 micrograms of lysin. Now, the lysin is delivered either nasally, which is not efficient. If we deliver it nasally, the enzymes will be aspirated into the lungs and hopefully treat. And this is just a contrast to treating it directly in the lung with a catheter, where we run a catheter into the lung and deliver the lysin directly into the lung. So you can see here the animals treated with buffer alone. Most of those animals basically died within 2 days. The animals treated nasally, this is not efficient. We can only save about 20% of these animals. Not enough enzyme gets from the nose into the lungs. But if we deliver the lysin directly into the lungs with a catheter, we get better efficiency, about 70% efficiency in survival. So I think we can get better and better at this by delivering the lysin to the lung where the infection occurs in order to kill the organism directly in the lungs. I'd like to say a couple of words about manufacturing. Lysins are produced in E. coli expression systems. Right now, they can get up to 1 gram per liter, very efficient system, but it is very expensive to produce. So lysins are really limited because of this expense to human treatment. But now we could produce lysins in food algae, such as spirulina, which is a food algae. If you go to Jamba Juice, you can get spirulina put in your juice as an added protein boost. So we can engineer spirulina with the help of Lumen Biosciences. This brings the cost down significantly, down to pennies a dose. Turns out that 5 to 15% of the dried spirulina biomass is the expression protein, is the lysin. So lysins can then now be fed directly with spirulina, or the spirulina could be put in capsules to treat intestinal infections. And we've done this. I don't have time to get into it today. We can allow lysins to be used in a veterinary field because we need low cost for those treatments. So we can use them in veterinary fields. And because they're so cheap, they can also be used for decontamination of surfaces. So using spirulina as a manufacturing tool, we can lower the cost of lysins and use them in areas of that we couldn't use them before. And you can see here, these are lysins, C. difficile directed lysin produced in E. coli and in spirulina. And you can see in various concentrations, you can see the optical density drop here is virtually the same as those produced in spirulina. So it's the same molecule produced in either E. coli or spirulina. So as the last couple of slides, lysins have been successful in completing phase 2 clinical trials. One of our lysins, the one I talked to you about, PLISS2, is directed against Staph aureus, has successfully completed phase 1 and phase 2 clinical trials to treat hospital patients with Staph aureus MRSA, bacteremia, and endocarditis. The treatment is a single IV infusion of the lysin plus standard of care antibiotic. So the single dose will reduce the bacterial load in those patients to allow the host immune system and the antibiotic to clear the remaining organisms. The lysin is now in phase 3 and should be completed within the next several months. So this is the first and only antibiotic alternative to achieve this FDA milestones. And I think it's a great achievement. And hopefully if phase 3 is completed, then it opens the door for other lysins to be used as a alternative to antibiotic. So as a final slide, I just want to talk about timeline for clinical development, because I always get the question, why, if you discovered lysins in 2000 and 2001, why did it take so long for it to get developed? Well, will point out what happens. So basically, the first 3 papers that we published on lysins were published in 2001 and 2002. The first confirmatory paper was published a year later in 2003, and this was a group in Spain that were actually working with lysins, and then when they saw our paper, they repeated our experiments and shown that they do work. We then published 3 other papers on lysins, and the second confirmatory paper was published in 2006, and this was a group that were never working with lysins. They started to follow our methods and produced the same results. In 2011, 10 years later, the lysin was licensed by a biotech company for therapeutic purposes. And 3 years after that, they received FDA approval to start clinical trials. So it didn't take very long after they licensed it to get approval for clinical trials. They started phase 1, phase 2, and now they're in phase 3. So this period, 10 to 14 years, is a pretty normal development time for any antibiotic. And this is a first-in-class antibiotic, so it's not unusual for it to take this long. So in summary, gram-positive lysins are a new agent that control gram-positive bacteria, are effective against antibiotic-resistant organisms, work synergistically with antibiotics, because I think it's one of the ways it's going to be used in the field, are difficult to become resistant against. We don't see resistance even over 20 years, even after trying to force resistance. Antibodies don't neutralize their activity, even though antibodies are produced in very low quantities against lysins in humans, they don't neutralize their effects. And based on phase 1 and phase 2 data, they're extremely safe. The gram-negative lysins, whole lysins work topically, but need engineering for systemic use because they become inactivated in serum. They work in the presence of lung surfactants, so we can use them in lung infections. And the charged peptide is more effective, but also needs engineering to work in serum. So we are now using and producing lysins from food algae, so the costs of the materials may be markedly reduced, and we can use them in other applications. So just like to acknowledge those people in red that I basically have told you about their work today. We've collaborated with several people with their various animal models, and we sent them enzyme for them to use them in their animal models, and worked quite well. And we're supported by a number of groups. And I must highlight DARPA. The DARPA actually put the most money into this early on to develop most of the enzymes that we developed in the early days. Happy to answer any questions that you have. Thank you.
Thank you very much, Professor Fischetti. This is an amazing journey of your lysins, and I think your research has paved way for so many researchers across the world to feel inspired and think of lysins as potential therapeutics. So thank you so much for this seminal work, I would say.
Thank you.
Yeah, so there are questions. I'll start taking them. So the first question is, I mean, I think that people have in general apprehension about bacteria gaining resistance against lysins. So I think that you've already answered. So I would skip that question. I had just one simple question. When you said that in the presence of surfactants, you found the enzymes to be working. So to me, the fatty acids in the serum, I read somewhere that fatty acids in the serum may possibly bind to lysins and hence reduce their efficacy. So does it have any relevance that when you add surfactant, you find a better result?
Well, we're not sure it's just a guess that the positive charge region is binding to some component in serum, because once we add serum, the enzymes are pulled out of solution. We're now we have a grant to actually look at this to determine why they're being inactivated in serum. So, but we don't know if it's fatty acids or whether it's albumin. All of those molecules are negatively charged in some local area, and that could be sufficient to pull out these lysins in those locations. But we don't know exactly what the reason is.
Thank you. I see Daniel Nelson also here. He's from your past team. Okay, so this is question, Professor Fischetti. How does the animal model immune system respond to the lysin molecules? I think this was given earlier, but would you like to take this question? How does the animal model immune system respond to the lysin molecules?
Well, the animals will respond in very similar way as a human would respond. The animal will make antibodies to the lysin if left long enough. But we've shown that those antibodies, we've made hyperimmune rabbit serum, and they don't neutralize the effects of the enzymes. The immune system will not. The only problem is that if you have a lot of endotoxin release, then you can heighten the cytokine induction and could cause issues. But we don't see any major problems if one uses a therapeutic dose of lysin, which doesn't break up the cell wall in lots of fragments, which could cause an inflammatory response in animal as well as human.
Vijay Kumar has a question, and he had a very good comment to make. So, I did not mention in the previous talk also. So there are a lot of complimentary messages for both the speakers for the excellent presentation. So a lot of people have said they're very happy to have attended these talks. So I want to convey that to you and to BG. And Vijay is asking, is there any difference in level of action with regard to ectolysins from endolysins?
exolysins and endolysins.
Actualizins, the license which are used for, the VAPG, the actualizins and the endolysins. Endolysins are secreted from within, and the actualizins are used by phages to inject their genetic material. Okay, probably. Okay, I'll skip this question.
I have to think about that.
Okay. What is the minimum bacterial count which can be treated in vivo using license?
You mean bacterial count?
Yeah. What's the minimum CFUs?
The minimum CFUs?
I think that's what this person wants to ask. Minimal bacterial count which can be treated in vivo using license, which means maybe the infection is not very severe, but the bacterial load is low.
The fewer the organisms, the more efficient the lysine. So if you have lots of organisms, you have much more difficulty in treating the infection. But if you have fewer organisms, it's very efficient to kill the organism, as long as you have a sufficient concentration of enzyme in the blood. But the fewer the organism, the more efficient the enzyme. Because the enzymes, as I've described, have a binding domain. And that binding domain binds to the lysine.
Right.
And once it binds, then the lysine will kill. So it's very efficient.
And now, since we saw in your slide also 20 years and no resistance. So what could be the reason that bacteria do not develop resistance against lysins?
Because they are essential for phage growth. If you think about it, if the phage, once a phage gets inside the organism, it has to get out to survive. If those phage are not able to get out, then that gene cluster, that gene clone of phage is lost forever. So through cycles of resistance, that a bacteria becomes resistant to a lysine, then those trapped phage are eliminated from the population until such time is that lysins that target certain receptors or certain targets in the cell wall that the bacteria can't change easily. Those are the ones that remain, and those are the ones that have remained now.
Okay. What are the chances of our body clearing the lysins from the system? Like once you administer, and what are the chances that it will be cleared from the system?
They're cleared. They found that the half-life of lysine in humans is about 2 hours. In a mouse, it's about 30 minutes. So you eventually clear the lysine. It goes out through the kidneys likely because it's small enough to go out through the kidneys. So you do clear it. You must. I think later on, after this lysine, a single dose lysine is accepted, we'll probably start using lysins in multiple doses because I think it's more efficient in using it in multiple doses. But because of the clearance, like antibiotics are cleared within hours, lysins are cleared within hours.
Right. Varun wants to ask, how specific are the peptides for the host, like the peptides that you showed in your slide? Do they have any off target? Do you expect them to have any off targets?
So far, we haven't seen any off targets. We haven't just tested all off targets. We've tested against red blood cells. We've tested against B cells and epithelial cells, and it has no effect on those types of membranes. Surprisingly, which surprised me as well, that these peptides, despite the fact that just charged regions, they work against the organism from which the phage work against. So, for instance, the peptide that was derived from a gram-negative lysin for Acinetobacter will kill Acinetobacter and maybe a few other organisms, but more efficient against Acinetobacter. They're not broad-acting peptides, which is surprising. It says something about the orientation of the charge regions in that peptide and the charge regions in the outer membrane of the gram-negative organism. So there is an association. We're trying to understand that association, but it's surprising that the peptides are fairly specific for the organism that they're designed for.
Right. Thank you. Prashant wants to ask, are license can they be used prophylactically? Given that in secondary bacterial infections, we give antibiotics prophylactically. So how about license given?
Prophylactically, in the sense that you can reduce the carriage of a pathogen that could eventually cause a secondary infection.
Right.
But you can't use it prophylactically for that organism that is going to cause an infection down the road, because you'll clear it out of your system. So it's the idea of removing an organism that could potentially cause an infection, as I mentioned, as I described in that model.
All right. Raza wants to ask Professor Fischetti, gram-negative lysins are not as specific as phages. So can these lysins be used in the gut where there might be some of the non-pathogenic gram-negative bacteria as well?
They can be used in the gut, but it could be more difficult in their use in the gut because of penetration. Penetration is a big difficulty in the gut because the organism needs to penetrate into the fecal material, and it's very difficult for it to do that. If it can treat organisms that are causing infection along the mucous membrane, then I think treatment could work. But I don't think it could penetrate into the fecal material to kill the organism in that location.
Right. So, so two related, slightly related questions. I'll club them. One is that can they withstand stomach pH when taken orally? And how feasible is the application of license to obligate intracellular pathogens?
Well, the intracellular, we haven't seen them going intracellularly. We haven't tested it formally, but we don't believe that they will get intracellularly, like the Phage Lysin, the only lysin that we know of that can go intracellular. The other question, the first question was
pH of the stomach.
Yeah, we've not tested formally, but I don't think it will be able to get through the stomach, but that's easily treated with bicarbonate. We've also placed lysins in capsules that can pass through the stomach and get into the lower intestinal tract. So we can avoid that fairly easily. And I think these molecules probably could make it through the stomach, but I think there are better ways of doing that.
Right. So there are several more questions. So Ranjan, how much time do we have, Professor? I always have this dilemma. Questions are coming. How much time do you have to?
I have a couple of minutes. I have another call. I have a call after this.
All right. So in that case, maybe again, the same thing which I did, that I can direct these questions to you if possible for you to reply them.
Sure, I'd be happy to.
All right. So I see. Okay, because there's several, so I can't take all of them, but great appreciation. And I think with that, we would like to wrap the session. It looks to me a little abrupt because it was going very well. I mean, it was literally like question answer was very smooth session. Excellent presentation, Professor. Thank you so much.
Thank you.
And it was a great pleasure to have you on this forum. BG, are you there, or has Dr. BG gone? Is he there? So can we thank? So okay.
It's not here now.
Perhaps he's left, but it would have been great if you both had a conversation. But so on behalf of all of us here, we have, so Konanika, are you there? Konanika, okay. So Ranjan Gupta, you might know, and Joe Campbell, so they were behind, they helped me a lot in organizing the session. And you know that they got in touch with you. So thank you for your consent once again. Thank you to all the participants for staying back. I mean, for Indian subcontinent, it's pretty late. It's like dinner. They have, I think, gone past their dinner time. But thank you very much. And I thank all the participants, and especially the students from my lab, Ritu Arora, who has sent the invitations to everyone. So thank you, Ritu, and everybody from your team to help us organize this session. A very successful one, I would say. And I'm glad that license have got a lot of, I mean, they know because, you know, the phages have the phages have gone a lot of press, I think. That's my question to you. Why is lysine not getting as much press attention in spite of being?
It boggles the mind because it's gone so far in the clinical trials. It's far more advanced than phage therapy, yet it doesn't get the same publicity as phage therapy.
So I hope that today we have been able to garner a lot of attention, and I hope that a lot of people would like to at least think about working on lysins. Far easier also. I mean, recombinant proteins versus the whole phage. I think
It's, you know, just the one last point. Phage and bacteria have been battling for 1 billion years, and the systems have been developed for no one to win. Phage are not allowed to win, and bacteria cannot win. Phage therapy is trying to force the phage to win. And until we understand all the systems that bacteria have to prevent that from happening, phage therapy will be very difficult to administer. It'll work, but it's going to take a long time to really understand all the systems that allow the bacteria to prevent phage from winning. And we're trying to make phage win in a system that's evolved for 1 billion years. It's going to be very difficult.
Professor Sanjay Chibber, are you there? Professor Sanjay Chibber, are you there? Because he's the one from India who has been working extensively on lysins. He's an emeritus scientist now.
Yeah.
Yeah. So I just want to try once, if you could have a minute. Professor Sanjay, are you there?
Doesn't seem to be.
Okay. So I just wanted to introduce him to you. And so thank you so much once again to you and everybody, and to Ranjan and to Joe and Ritu and her team from ANTC. Thank you very much.
Thank you very much. Thanks, everyone.
Thank you so much. Bye-bye.
Bye-bye.